June 1st, 2016. An article was released on Science News titled "Jumping Gene Turned Peppered Moths the Color of Soot." This is the latest information on our gene of interest, the cortex gene. By applying advanced technology to the classic evolutionary study of Great Britain's peppered moth population, researchers have found mutations leading to the crucial color shift in a gene called cortex that holds elements explaining the mystery behind these moths. Before Britain's industrial revolution in the 1800s, the native peppered moths blended well with the surrounding species of lightly colored trees in their environment due to the moth's speckled gray phenotype. However, once the Industrial Revolution's factory pollution dispersed soot into the environment, darkening the trees and the skies filled with coal smoke, the famous color shift in the moth population occurred through natural selection.
With time, predators killed off most of the light peppered moths because they were easily seen against the black soot-covered tree trunks. Then with mutation, generations of moths could increasingly express a black phenotype better suited for their darkened environment.
These mutated black moths blended in better than the original white pepper moths against the soot-colored trees. Natural selection caused these moths to become more prevalent in the British peppered moth population because most of the white pepper moths were killed by predators. Victorian naturalists noted the newly discovered all-black Carbonara form of the peppered moths as the light-colored typical moth, lacking the advantageous mutation, were easily eaten.
With decreased air pollution in the late 20th century, trees eventually lost their black soot coloring and returned to their lighter trunk colors. The black moths were now the ones that were easily visible to predators. Natural selection then caused a resurgence in the light speckled gray population of peppered moths, now that they have the advantage of camouflage. In present day, the black peppered moths are considered rare.
Let's take a look at the moth SNPs, insertions, deletions, and transposons. Transposons are movable sequences that can jump around in the pepper moth's genome and cause mutations by breaking or interrupting a gene. They contribute to genetic variation and evolution as shown in the moth studied. In the moth's target DNA, staggered cuts are made. The staggered cuts leave the short single-stranded pieces of DNA. The transposable element is inserted into the DNA. The single-stranded ends of the target DNA, the sticky ends, are bonded to the single-stranded ends of the transposon. DNA polymerase fills in the gaps of the single-stranded DNA. Replication of the single-stranded DNA creates the flanking direct repeats.